Dr. Maguire and Dr. Bennett will take some questions from the audience now. We have a whole lot of questions on the website. I see the most common one is if we see that the vision is reducing again, do we do this procedure once more?
So we don't have an answer for that, just because I guess it's a good problem. We haven't definitively seen in the phase 3 the trial any reduction in vision, and I guess part of the thing is, why is the vision being reduced? So when you create one of these blebs, when you inject under the retina, we're not actually injecting under the entire retina. That would require, that would be more of a risk, and it would require a lot more material. We try and treat the most relevant parts of the retina for day-to-day vision, the center vision, and things you used to walk with. So it may be that the areas that don't receive the drug aren't working quite as well, and you might be able to measure that as a slight decrease over time, because those areas aren't receiving the drug. But I don't know that that would be a good reason to go back and inject, since those parts of the retina really aren't used very much for vision, though we can still measure. But yeah, it depends what the reason the decrease in vision and why it happened. It's conceivable that we might re-inject or do additional injections if it was the case that vision were decreasing and we could improve things by additional injections.
Thank you, Dr. Maguire. We have some questions regarding the age or the range of age that patients are selected for the treatment of Luxturna. Also asking if newborn screening is possible, and also if adults can be treated.
So, Jean, why don't you take the newborn screening, and I'll talk about the age range.
Sure. Okay, so for newborn screening, yes, indeed, it is possible to do that. At present, newborn screening is not covered by insurance. However, it's possible to do that on your own, and that could, again, be done from a tiny blood sample or saliva sample, or if skin is removed for some reason, that can be tested. However, I would like to add that if the individual is found to have an RPE65 mutation, the current guidelines for delivery of Luxturna require that somebody be 1 year old or older, so that there is a large time window during which this testing could occur prior to a treatment.
So in terms of the age range, as Jean said, it's 1 year and older, which is it was thought that at that point the eye is large enough that the treatment could be administered with minimal increase in risk. And in addition, the nerve cells in the retina are done replicating, so there would be no dilution of the drug, into more and more cells as they divide. In terms of the oldest, it all depends. There are people that we've seen who are in their third, fourth decade of life who actually have a population of cells that are still there and can potentially respond. So again, it depends on. There is no upper age limit as long as there are viable cells that are left and can be demonstrated. Unfortunately, there are some people we've seen who are, in their fourth, fifth decade of life, and honestly, they just don't have any population of nerve cells left. And for them, to go through a procedure like this would just be, it doesn't make sense, and it would actually be no advantage to them at all.
Thank you, Dr. Maguire. So another question, I think quite a few questions on whether this kind of approach can be used to intervene in blindness conditions which are due to other mutations, and especially a research on CEP290.
So there is a great deal of laboratory research that is ongoing all over the world, looking at ways of intervening with other forms of retinal degeneration, including CEP290. The particular challenge with CEP290 is that the gene is very large, and so it does not fit within the cargo capacity of the adeno-associated virus, the AAV, that we spoke about. But there are some other strategies that are being considered, and including strategies of allowing this gene to fit into that small cargo capacity. So back to the original question, the answer is yes. If the gene can fit and it can be delivered into through the AAV to the appropriate retinal cells, it should work to intervene with the condition and provide vision. And indeed, proof of concept has occurred in multiple genes in animal models in the laboratory, and is forming a pipeline that hopefully will translate into human clinical trials in the near future.
Thank you, Dr. Bennett. I would like to read out a comment that has just been posted to both of you. It says, I'm so grateful for this groundbreaking technology and the people who have made this possible. I have a 10-year-old daughter who is at her 2-week post-op in one eye and 1-week post-op on her other eye today. We have already noticed big gains in dim light. She is seeing things in a whole new light and is so excited about her new perspective. As a mom, to see her point to the sky and say, I see clouds, nothing is more beautiful. So thank you, thank you, thank you. That's what she writes. So Dr. Bennett and Dr. Maguire, I think you've made many, many children happy, their parents happy, and 25 years of research on congenital blindness. That's awesome. Has finally brought in this approval of Luxturna. And thank you so much. We're almost to the end of our hour. Thank you so very much for your continuous, persistent efforts, your time, and sharing all this information with us. We'll also like to thank Tabby and Sarah for participating, and Tabby for those excellent questions for your group. And also thank University of Pennsylvania and Children's Hospital of Philadelphia and the audience. This talk is recorded and will be available on CureTalks.com. So please visit our website for details on more upcoming talks. Thank you and have a great evening, everyone. Thank you.
Thank you.
Thank you so much.
Thank you.